This is our Chapter 9 video introduction. This chapter we're going to be working on the patterns of inheritance and genetics and the way that we pass on traits from one generation to another. This is a continuation of the material we talked about in terms of meiosis, when gametes are formed. Remember, we talked about how it's a chance of which way the chromosomes are going to be lined up, which one that came from your mother or your father is going to end up in that gamete, in that egg or sperm. So this chapter, we're going to look at the combination then of that egg and sperm and what we could possibly get. So genetics, or the kind of genetics we're going to learn about in this chapter, is the study of heredity, the way that we pass things on from one generation to another. And we're going to kind of look at dogs. What's great about dogs is they have been bred for certain traits, and those traits are characteristic to their breed and based on their genetics. So what dogs naturally do, because they are a retriever or because they're a guard dog or a working dog, is all part of their genetics and the heredity that they were bred for. So if we look at the picture of these dogs, I want you to look at it and make some observations. What do you notice about the body shape of the dogs, the fur, their behaviors? How are they working together? What is it that they're doing? How are they working? These dogs are huskies, and they have been bred to pull sleds. This is what they do. Notice how happy these dogs are. This is what they want to do. They have a broad, deep chest that gives them a lot of power for running, and that's where the harnesses are attached to. You'll notice that they have nice wide paws that help to give them traction in the snow. There's also fur in between their paws that helps to protect the pads of their foot from the snow. You'll notice that many of them, not all of them, but many of them have dark coloring around their eyes, almost like a mask that acts as sunglasses when the sun reflects off of the snow. Notice how they're working in a team. This is also part of their breed. This is part of their genetics, the behavior of recognizing that they are all working together. This is what heredity is about. These dogs, what they do, what they are drawn to do, is based on their genetics because we've bred that into them. We purposely, we being humans, picked these dogs for specific traits and continue to breed them to manifest those traits again and again, generation after generation. That is heredity, and it's based on genetics. So heredity is the transmission of traits from one generation to the next. And kind of the father of this science is Gregor Mendel, and we call this Mendelian genetics. So Mendel was a monk who lived in the 1800s, and he worked in the garden. And he used math as a way to analyze the patterns of how pea plants in particular grew and how their traits got passed on. He didn't have fancy equipment. He didn't have a lab. He didn't have any kind of electricity or a computer or Excel spreadsheets. He made observations. He crossed plants. And then he analyzed the patterns of their outcomes. And based on that, he developed the genetics, the Mendelian genetics that we're going to talk about in this chapter. Here's a picture. It's his Facebook profile picture. So in the garden, he picked these peas for a particular reason. Garden peas are very easy to grow. They come in many distinguishable varieties, which we're going to talk about, looking at flower color, flower location, seed shape, seed color, height. These are all very distinguishable traits that could be easily observed on the pea plant. Also, they can be easily manipulated, as I'm going to show you in the next picture. So we see this is a flower from a pea plant, and you will notice here that there are male parts of the flower and female parts of the flower. We're going to learn more about that later in the semester. But the flower type of this pea plant allowed Mendel to manipulate them. What he would do, as I'll show you here in a little bit, is he would use tiny little scissors and he would cut the stamen here, the male parts of the flower, so that he could purposely put pollen on the female part from a specific flower. So this is really one of the key reasons why he used pea plants. Mendel kind of developed a lot of the terminology that we use in genetics. He talked about these characteristics, or these characters that were heritable features that could travel from one generation to the next. And a trait is a variant of that character. So we might talk about character in terms of height of a pea plant. And the trait would be being tall or being dwarf. Or the character might be flower color, and the color might be the trait might be purple or white. So all of the characteristics that Mendel looked at had two distinct traits or forms. And this is important to kind of the backbone of Mendelian genetics. He created what he called these true breeding plants that were kind of pure in the sense of that trait that he was looking for. And he would take two different pure plants and cross them and create a hybrid. A hybrid is offspring that comes from different types of parents, or the combination of genetics from true breeding parents. And he also instilled this concept of generations. These words that he used were the parental generation, the first filial generation, and the second filial generation. This is kind of how he kept track of things. And he kind of shortened it by calling it the P generation for parental, the F1 for the first filial, and F2 for the second filial. If we are comparing this to kind of our own personal history, then we might say that the P generation is the grandparents, the F1 generation is the parents, and the F2 is like the grandkids. So here's how he did it. Mendel took these tiny little scissors and would remove the male structures of one plant. Then he would take a paintbrush and brush the pollen off of the male stamens here from another plant, and then put that pollen directly onto the female flower structure. This allowed him to have control over fertilization. He purposely is picking a certain kind of sperm and connecting it with a certain kind of egg. So then inside of the female part here, there's an ovary, and that ovary gets fertilized. The eggs get fertilized inside of there from the sperm that come from the pollen and produce this pod. And inside of that pod are the seeds. So he would then take those seeds and plant them, and those seeds would grow into the F1 generation. So he tracked these traits, looking at these two alternative versions or traits, such as flower color, as we see here, purple versus white, flower position, whether the flower was axial, meaning it came off the stem, or terminal, it was at the end. He looked at seed color, he looked at seed shape, pod color, and stem length. Notice that all of these have two variants, two traits, one or the other. A purple flower or a white flower. Here's what the flowers actually look like, and you can see that this is a flower that would be easy for Mendel to manipulate. So he created these monohybrid crosses. Mono means one. So we're looking at one characteristic and the two different traits. And so all of the genetics that we're going to do are really based on a monohybrid cross. So here's what he did. He took that purple flower, that true breeding purple flower, and he crossed it with the sperm from that true breeding white flower. That's our P generation. Remember we said that those sperm fertilized the eggs, the pea pod grew in those seeds, he harvested those seeds and planted them, and that was our F1 generation. What I want you to notice is that all of the flowers in the F1 generation are purple. That's really important. So then he took these flowers from the F1 generation and he crossed them, again with his scissors and paintbrush, and created the F2 generation. And what we see in the F2 generation is a ratio of 3 purple plants to 1 white, I should say purple flowers, to 1 white flower. What's neat here is that we see white flowers here that skipped a generation but then showed up down here. These are the patterns that Mendel was trying to understand, and these are the patterns that he was able to explain through mathematics. So here he is working with the pea plants. Again, not fancy equipment. He's not inside of a lab, but yet he was able to fundamentally develop the modern genetics that we have today. From this information, he came up with 4 hypotheses for a monohybrid cross. First of all, he said that there are alternative versions of a gene, and he called these alleles. Remember, at this time, they didn't know about DNA. They didn't understand chromosomes. They knew there was a genetic component, but they really didn't fully understand it. So he termed these genes, or these versions of a gene, an allele. And he said that you have 2 alleles for each trait. You get 1 allele from mother, 1 allele from father. If those 2 alleles are the same, then you are said to be homozygous for that gene. The prefix homo meaning same. If the 2 versions of the gene, or the 2 alleles that you get, are different, then you are heterozygous. Hetero meaning opposite. He also noticed that the way these alleles worked is that one of the alleles always dominates over the other. That's not determined by humans. That's just the way the genetics works. So one allele is referred to as the dominant allele and is always represented with a capital letter. The other version of the gene is a recessive allele and is always represented with a lowercase letter. If you have 2 dominant alleles for a gene, then you would be considered homozygous dominant because both of them are the same and they're both dominant. If you have 2 recessive traits or 2 recessive alleles, then you would be homozygous recessive. Homo because they're the same, and recessive because they're both the recessive trait. If you have 1 dominant and 1 recessive, then you are considered heterozygous. You don't have to say dominant or recessive after that. You just say heterozygous. The last part of this that Mendel developed was what we call the law of segregation. And this really stems from what we learned about meiosis. And it says that each individual has a pair for that trait, for that characteristic, 2 alleles. In their gametes, in their egg or sperm, they can only give 1. So that pair segregates or splits, creating 2 different versions of a gamete. And this leads to the variation. This is how the white flower can skip a generation and show up in the next generation because of the law of segregation. Speaking of heterozygotes, so the way that we calculate the possible outcomes based on a person's alleles is using a Punnett square. And this is where I'm going to stop because we're going to talk about that together in class.